Hello, welcome friend. Welcome to our channel Quality Group. Friend, today our topic is Pharmaceutical Quality System, that is PQS, ICHQ10. Introduction about PQS. Foundation, Regional GMP, that is Drug Product Requirement, thus ICH Guidance Q7, Good Manufacturing Practices, Guidance for Active Pharmaceutical Ingredient, and ISO Quality Management System guideline form the foundation for ICH Q10. Harmonization, ICH Q10 provide a harmonized model for a PQS life cycle. It defines how a modern quality system assures science and risk-based drug manufacturing and quality decision throughout the life cycle. Significance of PQS. PQS establish and maintain a state of control. PQS facilitate continual improvement. PQS facilitate effective knowledge transfer and management. PQS facilitate implementation and effective utilization of quality by design, that is QBD. Q8 pharmaceutical development and risk management, that is Q9. 6. Quality system of PQS. FDA inspection program includes 6 systems: material management system, equipment and facility, production system, laboratory control, packaging and labeling, and quality system. Leadership and senior management. It includes quality culture, business case for pharmaceutical quality, management responsibility, state of control, innovation, root cause, life cycle, statistical analysis, process performance and capability. QC versus QA. QC, that is reactive, and QA, that is preventive and proactive approach. Continual improvement and variability reduction. Quality culture. It consists of support for the quality organization, action more than words, investment in quality, quality involved in relevant business decision. The quality of the work you accept become the organization's standard. Organizational structure. It assures that QA is independent and not subordinate to other organizational units. Quality culture. These are all integral to continuous learning and improvement. It includes supplier relationship throughout the life cycle, science-based approach, decision based on understanding product intended use, proper identification and control of area of potential process weakness, including raw material, responsive deviation and investigation system that lead to timely remediation, sound method for assessing risk, well defined and designed processes and product from development through entire product life cycle, system for careful analysis of product quality, supportive management, philosophically and financially. GMP is good business practice. PQS further align GMP with basic business goal of process predictability, example right first time, and product dependability. Dammings chain reaction. It includes reduce variability, improve quality, decrease cost, that is rejected good, etc. Better products and productivity. More competitive. Measuring performance is fundamental to any business. Actual performance versus standard. Identify process and product quality performance gaps and promptly correct root causes. Prevention. It includes preventing manufacturing problem is good business. Innovation. The introduction of new technology or methodology, ICH Q10. Introduction, implementation of ICH Q10 throughout the product life cycle should facilitate innovation and continual improvement. Change management. Innovation, continual improvement, the output of process performance and product quality monitoring and kappa drive change. Facilitate continual improvement. Implement appropriate product quality improvement, process improvement, variability reduction, innovations, and pharmaceutical quality system enhancement. Continual improvement of the PQS. Continual improvement of the PQS, innovation that might enhance the pharmaceutical quality system, knowledge management. Sources of knowledge include but are not limited to manufacturing experience and innovation. Awareness of new opportunity for innovation such as advanced technology, manufacturing, analytical. Significant opportunity exist for improving development, manufacturing, and quality assurance through innovation in product and process development, process analysis, and process control. Guidance for Industry Pact, a framework for innovative pharmaceutical development, manufacturing, and quality assurance 2004. Unit dose uniformity performed in process, example using weight variation coupled with near infrared NIR assay can enable real time release testing and provide an increased level of quality assurance compared to traditional end product testing using compendial content uniformity standard. Innovation, some examples of what process analytical innovation. Process analytical technology PAT can measure in situ or at line API process step endpoint, residual solvent label, particle size, identity, granulation, progress and completion, drying endpoint, blend uniformity, content uniformity, assay, weight, hardness, thickness. Determining the root cause of quality issues. Determining the root cause of quality issues, assuring quality each production day, each dose for each patient. Determining root cause. Two very common issues, two very common causes are human error and raw materials. Human error, cause of substantial variation across the industry. Human error can be prevented by analyzing the process for failure mode and increasing automation. Human error analysis. Human error training allow for deeper insight into the underlying causes of human error in order to identify and avoid its sources. Raw material, excipient, FDA recall, root cause, research finding, the global supply chain responsibility. All parties who manufacture include testing, process, pack, or hold an ingredient or drug product are responsible for meeting CGMP. Adulterated ingredient is equal to adulterated drug product. Recall, follow stringent recall procedure. Follow stringent recall procedure if any feasibility. Example of excipient variability. Excipient variation has resulted in several recent recalls due to poor dissolution. Tablet lots had significant dissolution failure due to variation in the coating agent. Soft gel capsule failed dissolution as a result of a cross-linking of short-chain aldehyde and other liquid components. Extended release tablet failed dissolution due to variability of ethyl cellulose excipient. Oral powder for suspension product failed dissolution due to glyceryl vanillin acid value. MCC morphology. Microcrystalline cellulose from new source was initially found equivalent to original source. Microcrystalline cellulose sample were later found to have meaningful morphological differences that lead to dissolution failure. Some of the MCC lot had round particles while others had long fiber. CGMP, every batch, every day. Lesson learned, do not underestimate the impact of morphological characteristics of dissolution. Reinforce complexity of dissolution, disintegration, attribute and importance of test. CGMP, every batch, every day. We rely upon the manufacturing control and standard to ensure that time and time again, lot after lot, year after year, the same clinical profile will be delivered because the product will be the same in its quality. We have to think of the primary customer as people consuming that medicine, and we have to think of the status and what we are guaranteeing in there, that the drug will continue to be safe and effective and perform as described in the label. Thank you, thanks a lot, and keep watching our channel. Like, comment and subscribe more and more. Thank you.